Hello everyone, welcome back to my channel. Today we will talk about production of glutamic acid. In my previous videos I have already talked about production of penicillin, production of citric acid and production of vitamin B112. So today we will talk about production of glutamic acid. Starting with the introduction, glutamic acid is an alpha amino acid that is used by almost all living beings in the biosynthesis of proteins. It is non-essential in humans. Why we are calling it non-essential? Because the body can synthesize it. It is also an excitatory neurotransmitter, in fact, the most abundant one in the vertebrate nervous system. It serves as the precursor for the synthesis of the inhibitory gamma aminobutyric acid, that is GABA. Now we will talk about the functions of glutamate. Glutamate also plays an important role in the body's disposal of excess or waste nitrogen. Glutamate is a key compound in cellular metabolism. Glutamate is the most abundant excitatory neurotransmitter in the vertebrate nervous system. Glutamate also serves as the precursor for the synthesis of the inhibitor gamma aminobutyric acid, that is known as GABA. Glutamic acid often is used as food additive and flavor enhancer in the form of sodium salt, and that particular sodium salt is known as monosodium glutamate, that is MSG. Oxygrow is a plant growth preparation that contains glutamic acid, and basically the concentration of glutamic acid in oxygrow is 30%. Now we will talk about industrial production of glutamic acid. Glutamic acid is produced on the largest scale of any amino acid, with an estimated annual production of about 1.5 million tons in 2006. In 1950, chemically synthesized by the aerobic fermentation of sugars and ammonia in the 1950s with Corynebacterium glutamicum, which is also known as Brevibacterium flammum. Isolation and purification can be achieved by concentration and crystallization. Now, production of glutamic acid. For the inoculum, different strains are used for the production of glutamic acid. For example, Corynebacterium species, Brevibacterium species, Mycobacterium species, Orthobacterium species. Now we will talk about the media which is used for the production of glutamic acid. So glucose is used as a source of carbon. Ammonium acetate is used as a source for nitrogen. Corn steep liquor is used. Enzymatic casein hydroxide is used. dipotassium phosphate, magnesium sulfate, urea, and water is used. And bioparameters like pH, temperature, aeration, agitation are also required to enhance the yield as well as to maintain the process during the production of glutamic acid. So the pH has to be maintained near about 7.8, temperature 38 degrees Celsius, aeration a continuous steam or sterile air is pumped into it. For agitation is achieved by turbine type impellers for constant agitation, and normally the time period is 2 to 4 days. Now, industrial fermentation process is carried out in three different ways: batch fermentation, fed batch fermentation. The fermentation process completes within 2 to 4 days, as I told you earlier. In the end of fermentation process, the broth contains glutamic acid in the form of its ammonia salt. Now, talking about the recovery of glutamic acid, in downstream process, the bacterial cells are separated and broth is passed through a basic anionic exchanger. Glutamic acid anions get bound to the resin and ammonia is released. The ammonia can be reused in fermentation. Glutamic acid is crystallized and dried to store for a longer period of time. Now, talking about the uses of glutamic acid, glutamine is an energy source for intestinal and immune cells. It also helps to maintain the barrier between intestines and the rest of your body and aids with proper growth of intestinal cells. So, all these were about glutamic acid, about its production, about its recovery, about the different steams used, and also the uses of glutamic acid. Hope you understood about today's presentation. If there is any query, any questions, you can mail to me, you can comment also. Thank you.